Yep.
Okay, great. Okay, great. So the title of the talk is Space is a Sequence and Unifying Time and Space in Cognitive Maps. And it's based on this paper that we have put on bioRxiv, which is now accepted for publication. Hopefully it will come out in a couple of weeks. And we have another new paper that will be coming out soon because we have even more results. So before I dive in, I just want to give you a context on why we are interested in cognitive maps as a company, because cognitive maps fall on the path to building general intelligence, because it is involved in concept formation, schemas, situated language understanding, episodic memory, and navigation. So that's why it fits into our agenda. And if you follow our work, you have to. It's hard to connect the dots from outside, but we have been systematically building a cognitive architecture towards general intelligence. If you look at the publications, the first one was a generative model for perception, which Pratt captures. It is a structured generative perception model, which is used in a model called schema networks, which is for dynamics, learning dynamics and interactions between objects in the world. And another paper in 2019 was about concept learning and abstraction, and it uses an architecture that has all the other pieces in it. It has the generative perception model and dynamics model in it, and it is using these ideas from perceptual symbol systems, Barcelov's work, to form concepts and abstraction. And cognitive maps is another piece in the puzzle. Of course, it doesn't mean that we have put all these pieces together all in the right way, but that's what we are trying to do. And so it fits into that framework. So the agenda today is much less ambitious. It's just going to change the way we think about hippocampus and cognitive maps. That's the agenda today. And in fact, cognitive maps and place cells have all been about location. And I'm going to argue that that's the wrong way to think about it. Cognitive maps can be understood without thinking about space. And place cells is the wrong description. I'm going to try to convince you. And in fact, I'm going to argue that you should think about cognitive maps as pure sensation action sequence learning, nothing else. So space is a sequence. And all these disparate phenomena that we see in the hippocampus are all the same. And that's what I'm going to show. Boundary vector cells, landmark vector coding, splitter cells, sequential order cells, route cells, time cells, overrepresentation of the boundaries and rewards, lab cells, event-specific representations, place cell repetition, global, partial, RAID remapping. All of them can be explained pretty much using a simple idea of, in our model, which is clone-structured cognitive graphs. So let me get into what that is. So this is about learning sequences. Suppose you have sequences A, B, C, and DB in the world, just two sequences that you want to learn. If you try to model it using a first-order Markov chain, then what you get is this. So you have A, B, C in it, you have DB in it, but you also have unwanted sequences ABE and DBC in it, which were not part of the world. So it is overgeneralizing. So if you just look, these are higher order sequences, but if you just learn a first order Markov chain, it will overgeneralize. How do you overcome this? There is a trick called cloning trick. So what you do is, you are losing the context at B. At B, you are losing information from where did you come from. So you can create two copies of B and B prime, one corresponding to coming from A, the other corresponding to coming from D, and then relearn these outgoing connections to wire them up correctly. This is called the cloning trick. And one way to do this is to first learn a first order graph and find out where are the places in which you are losing higher order information, split that state into two, continue learning. So this is called a greedy splitting mechanism for learning. But that it's okay, but it's not great. We will describe a much better way of learning this, in a few slides down.
Dilip, I'm really sorry, and if you're going to get to this, don't worry. Could you go back one slide?
Yeah.
Can you explain the you mentioned after the next thing.
Yeah.
Yeah. So can you explain just with the cloning trick, what's going on with the dashed lines?
Oh, yeah, yeah. So suppose you learned this graph, and you found that, okay, I'm losing context at B. I decide to make two copies of B, one for incoming connection A,
Yep.
and another for incoming connection D.
Yep.
Now, but you don't know what the outgoing connections should be. So what you do is you copy the outgoing connections into both of them. So B and B prime will start initially with the same outgoing connections. The connections which existed here.
So you've learned ABE, you've learned DBC.
In this graph,
You
as a
have essentially what look like two episodic memories. So you have
Correct.
one episodic memory for ABC, one for DBE, and that's the cloning trick.
Correct.
And then but what's I'm still a little stuck as to what's going on in the B versus B prime. Maybe it's because I'm thinking about it as two episodic memories.
Correct. Yeah. So, but remember that, yeah, you can, I can come back to that one, but think of your learning a Markov chain of this. That is, you are just connecting state A to state B,
yep,
and state B to state C when ABC happens.
Mm-hmm.
And you are just connecting state D to state B, and state B to state E when
Mm-hmm.
DBE happens.
Yep.
If you do that, you end up with this one,
yep,
this graph. But now, in this graph, you also have sequences which are ABE, which did not occur in the world.
Yeah.
So that means this model is mixing up things. It doesn't know whether, when it is at B, it doesn't know, I came from A, so I should go only to C, which is what the world says. But this graph does not have that information.
Yeah.
So to get that information, what you want is, you want to keep the separate context.
Yep.
And that's why we are copying this, B. One B corresponds to coming from A.
Yep.
Another B corresponds to coming from D.
Yep.
Both are Bs, but they're coming from different contexts. Mm-hmm. And then if I'm just copying this first and cloning, then the outgoing connections initially I'll have to copy it from here. But then I know that by observing the world again, I know that when I'm at B prime, it is coming only from B. So sorry, when I'm at B prime, it's coming only from A, so it should go to C. So these connections will get thrown away.
I see. So the only thing that was confusing me was the dashed lines. So I thought that there is still some implicit.
No, so it's basically saying that these connections will, when you learn further, these connections will be thrown away, and you'll be left with two sequences which are A, B prime, C, where B prime is still the observation B, and DBC, DBE, where B is still the observation B, and these two will be clones of each other. This
is a lot like the sequence disambiguation stuff, like Eichenbaum did, and I get it now. I'm on.
Yeah, exactly. Now, going forward, so this idea of cloning, it's actually a pretty old idea. It's a reasonably well-known compression algorithm called dynamic Markov modeling, introduced in 1987 by Cormack and Horspool. And interestingly, this idea got rediscovered again, in 2016. It was published in Science Advances, without realizing that this connection exists, and they talk about creating copies for modeling context, and this cloning trick is creating so you are not just accumulating observations to create higher order state. You are creating a latent state. It's that B prime that you're creating is in a hidden space. So it is it's much more flexible and powerful than just trying to do n-gram on the observations. If you just create, let me create a state which is a concatenation of A and B, that is very different, which is in the observation space, which is very different from this cloning trick, which is creating higher order Markov chains in the latent space. And so now I want to just give you a mechanistic understanding of what happens in a clone structured graph. So assume that you have learned a graph which has this cloning structure in it. In this picture, all these states or neurons that fall into the same column are all clones of this observation A. And similarly, these are clones of observation B. Just ignore how it got learned. Suppose these connections got learned. So that means in this graph, where the sequences I am storing are, there's a sequence ACE, which is this. There's a sequence AB, COD, EEA, which is A, B. It goes to either COD and then goes to EEA. So that's so you see where the sequences got stored, how the sequences are stored in this graph.
Good. A question for you.
Yeah.
I'm sorry I keep interrupting, but I just want to.
That's fine. Yeah.
So how is it in the AB and then C comma D example? How is it it's not creating clones of ABC EEA versus ABD EEA? How is it they're being integrated? Is it just the case that C and D appear at the same time?
Oh, right. Exactly. Yeah. So in this case, yeah. So it's basically, after B, C, so the next state can be C or D. And that's basically, and their C and D will be occurring at the same time, or same relative position after the occurrence of B. That's basically.
So every time you see like a repeated observation that doesn't match the overall chain, you clone.
Correct.
Okay.
So now I want to show you the inference dynamics in this one. So suppose after having stored these sequences, I observe the sequence ABDE. So when A comes in, so that is this observation coming in, it will activate all the clones of that observation. There is a bottom-up observation which is activating all the clones. And then, of course, these outgoing axons will send their activations to their next whatever their connections are, the downstream neurons. So those are the activations just coming and waiting there. You can think of it like that. And now let's say the next one is B, and now the activations here are a combination of these bottom-up activations and the lateral activations, and the ones which have lateral activations get strengthened and they will propagate forward, and the ones which do not have the lateral activations, they will get killed and they don't propagate forward. And so these things will propagate forward. And now if the next thing was to come in was C, this would get activated. But in this particular case, it was D coming in. So this thing gets activated, and so this one doesn't propagate forward further, and this one will propagate forward. And so you can see how states propagate forward in this graph. Now I want introducing some terminology that I will use. So these connections which I did not show in that picture, that exists. So basically all these neurons are directly connected to these observation neurons. So these are the bottom-up connections, and these I will call these lateral connections. Those are the things propagating activities from the previous time instant. Now, and the activity of these clones at any time instant is a combination of the bottom-up observation at that particular time instant and all the context coming in from the lateral connections of all the previous time instants, and the total activation is a normalized version of bottom-up activation plus contextual activation. This simple formula works out right in the for the probabilistic calculations when they are done in the log domain. So it's although I did not use any math notation here, this is mathematically correct when you do the probabilistic computation. It's just bottom-up activation plus contextual activation, a normalized version of that. So I want to also point out that you are always dealing with graded inputs. In the previous animation, I showed it as B coming in. I'm 100% certain that it's B, but in real life, it is not like that. When B comes in, there might be some noisy activation on E. That's okay. That will just get accumulated into the graph and it will propagate correctly just using the same computational formula that I showed earlier. And suppose when D is supposed to come in, D did not have the most strength. D was weak, and a distractor symbol A was the one which was stronger at that time instant. That is fine too, because even if A activates everything, there is no context to propagate it forward. Only D has the correct context. So the D will propagate forward. And even more, suppose D was completely absent at the time and only A was present. even there, the model will propagate it forward. This is the idea called smoothing. That is, you don't assign zero probability to anything in the model. Like, so you basically say, even when D is not present, you will assign a small probability for D being present. And if that small probability exists, then the context will bridge you over. Even though the D did not appear, this state will still become active and it will propagate you over, because that is better than something appearing out of context. So just wanted to bring that out, saying that you are actually doing probabilistic inference in this model, and it will take care of noise and missing information, etc., in the appropriate way. And this can be implemented in biology using very, very simple ideas. So now, so there are advantages of clone structured graphs, but let me skip over that. It's just a list. It will become clear as we go. And then there's a question of, okay, how do you learn these clone structured graphs? I told you, oh, look, if these sequences exist, this is how we do inference. And this was one of the insights in our paper. The old ideas was, you can use greedy splitting and, start with the first order graph, split, 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 keep going like that. But it doesn't always work because sometimes you also want to merge. You don't want to split. You want to merge things together that you had split earlier. Imagine the world is just first order, then you will keep splitting and splitting, trying to discover higher order information and to fill up the capacity. So you don't want to be greedy with splitting. You want to have a flexible mechanism which will basically take an allocated capacity of clones and see how to use it optimally to model whatever the input sequence is. And this is where we use this idea from hidden Markov models. This cloning structure can be it's basically a restricted version of a hidden Markov model, and you can instantiate in the hidden Markov model as an initialization, and you can learn using the hidden Markov model's learning dynamics, and it works much better than just learning it using the old greedy splitting idea. And it can be combined with greedy splitting to make it better. We haven't done that yet, but that's some work in progress. And we have also played around with other ways of doing this, where you allocate the clone structure, and initially you start just memorizing the sequence, which is just allocate the clones as you go and just memorize the sequence. And then when the capacity starts filling up, start compressing it. We have a paper, it's called Memorize Generalize. It's another way of learning this clone structured graphs, and it has some better properties than this hidden Markov model idea that we published. But this is good enough to show almost all the things that I want to show in the subsequent slides.
I'm so sorry.
Yeah.
Can you go back just a second?
Yeah.
So how does the cloned HMM deal with the idea of not wanting to create a zillion units of A and B?
So what you're doing,
problem, right? That we're trying to solve.
So what we're doing is we allocate a capacity upfront. We basically say, look, every observation gets 100 clones or 200 clones, whatever it is. You pick a number and allocate it, and basically now tell the algorithm, use this number whatever way you want. If you want to use up all the clones, great, go and use up all the clones. If you want to use fewer of them, and what we have found is that in general, just over allocating the capacity works fine, and then it doesn't end up using all the capacity. It uses more clones where it is needed. It uses less clones where it is not needed. But allocating that capacity puts a pressure on the algorithm to not just greedily, keep increasing the number of clones. That's basically it. Yeah.
Okay, but then the hidden Markov model, are you just greedily increasing the number of latent states?
So in a hidden Markov model, if you do it in just a standard hidden Markov model, it won't work. So you need to put in this cloning structure as a prior on the hidden Markov model. So that is a prior on how the hidden states of the hidden Markov model connect to the emission states. If you instantiate just a hidden Markov model and just increase the number of states and try to learn it, it just gets stuck in a very local minima and it won't learn. So you need this cloning idea to make it work. And then if you just give it enough capacity, it will learn the right thing.
Okay.
Yeah.
And is this somehow related to a POM-DP then?
Yes, it is a POM. You can call it
subset of a POM-DP, restricted POM-DP or something.
It is a restricted POM-DP because
okay.
Because we also have actions. Whatever is taking from this state to this state,
yeah.
There is an action label here. So okay. You're right. It is a restricted POM-DP. Yeah.
Okay.
Yeah. And one question that we got during the review of the paper was that doesn't HMM and EM learning make this non-biological? Not really, because EM learning is very similar to HTDP. Wolfgang Maas has a paper on this one, and the inference I showed you is pretty biological. It is, it's a message passing algorithm. So both are very biologically plausible. So we have references in the paper pointing to that now. Okay, so now let's dive into what can this thing do? And
before you jump into that, just one quick other question is another way of sort of thinking about this space is in terms of a distributed representation. The clones are a distributed representation.
Yes.
And so if you have an LSTM or other kind of recurrent neural network, is that kind of similar? I don't know if you've thought about, specific contrasts, what is the essential difference between this approach and an LSTM or other recurrent neural network?
So one is handling of uncertainty.
Okay.
Second is having that actual graph to reason over. So you are not just trying to, in our setting, once you have the model, you can reason back and forth on that graph. In that LSTM, you're just trying to predict the next step, and also it's not uncertainty aware. When you're LSTM, it's not an uncertainty aware prediction, right? So handling uncertainty, reasoning about the structure, all those things are missing in that one, which is there. Yeah.
Okay.
Cool. All right. So now jumping into the results. So the first one is spatial representations from purely ordinal sequential experience. So imagine you have a room, and the room has only 4 distinct observations. So the color is the observation that you're getting. When you are in this part of the room, you see pink. Here it is blue. Here it is green. And and now you are an agent walking in this room now. You're dropped in this room and you're taking random walks. And the sequence of observations you see are this, right? And of course you see your own actions you're taking, which are 4 different actions. And then if you learn a first order sequence model on this one, the Markov chain, this is the thing that you'll get. It's not a good model of this sequential experience that you had there. If you use the CSCG, that's what our clone structured cognitive graphs, that's what we call our model. If you use that, it will actually recover the room. Exactly. And remember, there is no Euclidean assumptions in this model. It's just purely sequences coming in, and it recovers the room layout exactly. And even better than that, suppose the room is actually empty in the middle. So you have some edge observations here, which are just denoted by color here. The corners are unique. And now if the agent is walking in this room, it's a pretty hard sequence. It's basically saying, okay, I got purple, pink, and then blank, 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 blank for a long time, then again something else like that. So it's a pretty hard sequence. This is a first order graph. Even in that case, the CSCG is able to recover the layout of the room. And this one degrades. As you increase the size of the room, the recovery will smoosh together the middle parts of the room. And the periphery will still be recovered more finely compared to the middle of the room. And this will appear as, oh, in the middle of the room, the receptive fields are bigger, and in the periphery, the receptive fields are overrepresented, etc. That's the interpretation that you will, if this is what is happening, that's the interpretation that will you'll come up with when we are measuring the place fields. In fact, that's what I'm going to look at next, about a quick walkthrough of the mechanics here. So remember that that room, in that blank room, what we are seeing are 9 different observations, and we are setting up the network to have some number of clones, and then so we have 9 different observations, and input at every instant is just a 1 hot 9 vector with 1 observation present. I'm saying, oh, it's a color present, right? That's a observation, and each observation is hardwired to the clones of the same color. That connections are not drawn here, but you should just have to imagine it. And the lateral connections between these are initially random. So imagine just lines going everywhere. And remember that where this highlighted observation, which corresponds to this brown color, it can be anywhere in these places. When the agent is here, this thing is active. When the agent is here, it's just the same observation active, etc. There is no location being sensed. The learned lateral connections after learning through our HMM will look like this, and which is the same as this. I just removed the dead neurons. And if I just rearrange the connections there without changing any connections, it just looks like this. And it's the same graph. I just draw it like this. So but you have to keep in mind that the graph does not have any length or width or anything like that. The graph actually looks like this. There is no width of the room in this graph. There's no length of the room in this. There are no locations in this graph. It's just a connection between sensory observations and some latent states. No idea of location in this one, right? And now, I want to point out that this is very different from where many other models start. For example, if you look at success representations, there is this thing called states to states thing. But in that thing, the states are actually locations. It's as if somebody is inputting locations in the model. I don't know where that locations will come from, because input is sensory observations. It cannot be locations. If the locations are directly sensed, there is no problem to be solved, frankly. So that's a difference. Although you draw these matrices as states to states, in that model the states are actually locations. So that is whereas in our model the states are just hidden states of observations, and the inputs are just observations. Important distinction to keep in mind.
All those standard reinforcement grid world kind of models always build in that assumption, right? So
exactly. So and then
very tricky that's like the convention. So it's like that's the problem to be solved. Yeah.
Exactly. Yeah. Exactly. Yeah. And so it's basically once you sense the location, there are no interesting problems to be solved.
Exactly.
Then you have to just kind of make up into problems to solve. That's okay. So good that we are in agreement on that one. So so coming back to this one, there is no width or height in this graph. There are no locations in this graph. But we as experimenter on this rat that has ran in this room can ask this question: hey, when does this neuron light up? I can point to a neuron in that graph and say, when does this neuron light up? In which place in this room does this neuron light up? And that is what we call place fields. And I'm going to argue that all the phenomena that we observe are an artifact of measuring place fields like this. And I'll show you why it is. So let's say we want to ask, what is the place field of this neuron? So the way we do that is we let after having learned the room, the agent has this graph in its brain, and we let the agent run in this room, and we basically ask, we mark the locations where this neuron becomes active. When the agent is in this location, if that neuron becomes active, we will put a point there saying, okay, there is an activation there, and we get a place field. This is the place. This is how we derive the place field. So, and that neuron that I showed in this picture is this neuron. I just rearranged the graph in this picture. It's this neuron. And yes, its place field is this one. That makes sense. Now we can ask the question: what will happen if I take this rat and put it in a bigger room? So the rat learned the graph in a smaller room. I take this rat and put the rat in a bigger room, and I'm asking the question: what will happen to the place field of this neuron? So don't let the layout of this graph confuse you, because now you have to think, you'll think, there's a width, length, etc. in this room. No, you have to think of, this is the graph, I'm just asking the question, what will happen to the place field of this neuron if I place the rat in a bigger room? And then, so this is the graph. This is the original place field. Does anybody want to answer this question? I'm going to let the rat run in this room and measure the place field using this learned graph. This is the original place field. Okay, I'll give the answer. The place field will become bimodal and expand out. And I'll tell you why this is. And this is the boundary vector self phenomena. I'll explain this to you in a few slides down. So if you expand the room in this direction, the place will become bimodal, and we will call it remapping. The place field remapped. But the mechanics are pretty simple, and it has nothing to do with boundaries or vectors. So remember, in this graph, when does this neuron become active? It's when it will become active along any of these trajectories. Because when the rat is at the boundary, it's not bound. It is more. It has a unique observation here compared to the one in the center. The unique observations are kind of anchoring points. If it is in the corner, that is a unique sensation. That is more of an anchoring point. And it doesn't need to be unique everywhere. Because of the latent space, it becomes unique as the rat walks around.
I had a quick, clarifying question. Why are there two yellow?
Right. Yeah, good question. So sometimes this learning doesn't recover perfect graphs. So sometimes it keeps two copies of the things around. In fact, I will go into you don't need to recover. You can keep some copies of the things around. So it's almost like I take a particular state and split it into two. It does not matter, because as long as both of them are wiring to the same destination, it is fine. It doesn't change the underlying representation or the probabilistic computation. So sometimes our learning plus the memory consolidation leaves some things overrepresented. Like some clones do not merge properly, leaving some imperfections, but it doesn't change the underlying representation at all. Does that make sense?
I think so.
Okay. All right. So ignore just assume they're the same for the time being, and because both of them will have the same input connections and same incoming connections and same outgoing connections, just divided by 2. That's what it's like. It's basically taking a neuron and dividing it by 2, and the incoming connections will be divided by 2, outgoing connections will be divided by 2, and which doesn't change anything. I was saying, this neuron will become active in these sequences when the rat is passing through here. That's what the graph says. And now we have put the rat in a different room. What will happen is that when these sequences pass through here, this neuron will become active. That same neuron will become active. And when the rat is going from here, that same neuron will become active, because the rat doesn't know that it's in this neuron. It's just it is working in the space of just the sequential observations I'm getting. Right? When I am here, if I saw blue and I took 5 steps like this, I am expecting to see this neuron fire. That's what the graph says, and that's what will happen when you do inference on that graph. So that explains this place field remapping when you expand the room. In fact, you can see that this is if I expand the room like this, it becomes bimodal in this direction. If I expand the room in this direction, it becomes bimodal in this direction. And if I expand both the width and the height, it will produce 3 different fields extra. And this is called the boundary vector self phenomena. But there is no distance computation or boundary vectors or anything in this one. This is purely sequence learning and just sequence inference.
So just a sec, that makes sense to me if you approach it from one of those other borders. But if you were to, for example, start on the left and just keep going, past the point where previously you had kind of connected those gray guys, what happens? It gets into this kind of weird place,
Oh, right.
like, you know, in the right-hand side, you're starting from the light blue and you just keep going into the gray territory. Now you've exceeded the normal sequence length of gray. What does it do?
Oh, because so the way it happens is that, remember, there is smoothing in the graph, right? Like, you know, everything is uncertain. So basically, this is what will actually happen in the model. The rat starts walking from here. It thinks it is, you know, it should be here in the corresponding room, right? And let's say the rat is actually trying to go to this green sensation, right? So at this point, okay, it says, okay, go forward, and at this point, it should be green. When it reaches here, it should be at the green sensation, but it is not. So it basically says, oh, I got a noisy observation. So my hidden state must be uncertain. So what should the next action be? That if I want to get to the green state, I should just still keep going forward, right? So what the rat will basically internally it will do is it will reset. It's basically thinking one step backward, basically thinking, oh, I made an error. Let me step one step backward and keep going forward. And this is what will happen at every time step, and it will eventually reach the place it is. So even though it is a even though it is using the wrong graph to navigate in this room, because of how the probabilistic computations are handled and how smoothing takes care of noisy observations, it will actually reach the place it wants to go to using this imperfect map.
Okay.
And this will also explain some of the lap neuron phenomena that Chenson observed. Like, it's basically this, when an uncertain observation comes in, the rat can basically internally adjust its thinking of where it is based on the probabilistic evidence it has. So it just thinks, oh, maybe I just made the wrong step, or maybe I saw the wrong observation. Maybe it's noise. And under those circumstances, what is the right action to take? And the model doesn't go backward. Model will say, still keep going forward. So which is the right thing? Okay, yeah, that's a very good question. And in all these, in our model, we are actually letting the model make the decision. It's not like we are showing this recipe fields. No, the model is actually doing the action and taking the decision and doing behaving by itself to do the right thing. Okay, so this is I think it's a classic paper from John O'Keefe. This is the result basically that I showed. This is the original room. When the room is expanded this way, it becomes bimodal. And when it expanded this way, it becomes like this, etc. So this is the boundary vector cell phenomenon. And I think this model captures almost all the phenomena seen there. In fact, they also show that the boundary vector cells are directional, in the sense that if you do the mapping only when the rat is moving rightward, you will only see this field. And if you do the mapping only when the rat is moving leftward, you will only see this field. That makes complete sense, right? So that's what exactly happens in our model. Basically, yeah, if the rat is moving both ways, you'll see this field and this field. But if it is moving only this way, yeah, you will only see this field. And if it is moving only this way, you'll see this field. And what you're seeing is a combination of both of them. So that makes complete sense. And again, no distance coding, no vector coding, no boundary, anything, just pure sequences. So that's, and the same thing can explain this landmark vector cells too, and if you don't remember this one, landmark vector cells is basically where, imagine you are in an empty room. Now you have, in addition to the room blank stuff and, observation, there are some objects in the room. There's one object here, there's another object here. And the rat is running and learning in this room, okay? And I want to see, suppose I found a neuron which has the place field corresponding to this location. So I found in my graph a neuron with a place field which is at this location, learned in this room. Now I am asking the question, what will the place field of that neuron will look if I move this object to this location while the rat is running, after having learned with object in this location? And this has been people have tested this one. I forgot to put in the reference here, sorry, but people have tested here, and what they found is that the neuron will open up a new receptive field here. And that's exactly what will happen in our model. It will open up a new receptive field here because thinking that because when it's almost like the boundary moving, or you are moving one observation. So it's the same phenomena, basically saying that when it is in relation to this one, it expects this neuron to fire, and then we are averaging over all those trajectories, so it will look like, oh, it opened up a new place field here. But it's not rewiring the connections or anything. It is just our way of interpreting what happened.
Can you just clarify, Dilip? Does is the algorithm, the model, just taking a random walk? Or have some policy?
No, just random walk.
It's not interested in objects or anything, which is something that's often been suggested with respect to these object vector cells.
So yeah,
objects.
Right. So in this model, I am only taking random walks. In the paper, I don't remember actually whether it was random walks or. So I'm pretty sure that the rat is not taking random walk when the object is moved. It will definitely pay more attention to the moved object.
It'll tend to approach it.
Correct. But it probably won't change things much in the argument, because what the rat is doing is basically when it is getting an expectation mismatch, it is exploring. One, of course, when it gets an expectation mismatch of the object should be here, it will explore. And when it sees a new object where it was supposed to be blank, it will, of course, pay attention to it and adapt its state, right? So both those things will happen. So I don't think it is possible to make a rat do random walk. Maybe you can put rewards distributor, etc., and it will do it. But probably it won't change the core of the argument in the experiment. That's my thinking. But yeah, it's something I should look into definitely.
And would your model accommodate changing the object? So what would happen if?
I see. Changing the identity of the object.
Mm-hmm.
Yeah, it will be just a. Won't be any difference between changing the identity of an object and changing a sensation. It's basically saying I can change the carpet in the room, or change, put in a new order in a place. All of them will look the same to the model. Does that?
I thought the hippocampal phenomenon depended on the identity of the object.
So I will go look into it. Like when you say the identity of the object, so if you put in a new object, that it won't remap, right? That's what you mean by depend on the identity of the object.
Yeah, exactly. So
that makes sense, right? Because what happens is that this object, the observations corresponding to that object, identity of the object is the observations corresponding to what that object represents, right? The sensations corresponding to that object got wired into the graph. And so if you introduce a new object in a different location, it will produce different sensations compared to what this object would produce when it is moved. Does that make sense?
I think so.
Yeah, okay.
Just curious how your model handled it.
Yeah. No, I should dig a bit deeper into that to figure that out. Yeah, cool. Great. So this is a experiment in the paper, and it's a pretty easy to understand. Suppose we know that hippocampus is involved in transitive inference, and the typical example shown is if you see A greater than B, you also see B greater than C. Can it infer A greater than C? This is the transitive inference phenomena that, or some versions of that, right? We can do much, complex versions of it. So imagine that there are two rooms, and they are again aliased observations. The observations do not identify the locations, and the rat runs exclusively in the first room or the second room. Never crosses between the rooms. So, but the rooms have overlaps, and not only that, the overlapping portion. The sensations in this portion also appear here. So it's like a piece of carpet which repeats here and here. Now, so this is pretty hard to recover the layout of the room while not confusing this portion with this portion. But our model is able to do that. And now what will happen is that if you, suppose after having learned this, if you put the rat right in the middle here, it will not know whether it is in this location or this location, because the sensations are exactly the same. So what will happen is that as long as the rat remains in here, it will have both these neurons active. And as soon as it steps out of this room, this portion will shut off, which is because that is corresponding to this part. And then when this portion shuts off. And then when the rat comes back to the original place, only one of them will be active. The other part will not be active, because now the rat knows that, okay, I am in this, because just stepping out is enough to resolve all the ambiguity. So basically saying the rat is getting the same input sequences from, so this input sequence is X. The activity is different, because by the time the rat comes to X again, it's in a different mindset already. So that's why the activity of the clones is different. So this is another set of, I think, classic experiments from Muller and
sorry, Dilip, I might have missed something sort of fundamental there, but how is that the same as transitive inference?
Oh, yeah, transitive inference. Sorry. So I should say transitive learning. So in this case, the rat. So the transitive inference is almost trivial after having learned this thing in our model. So it's basically this. Suppose the rat ran only in this room and this room, and it recovered this graph, right? Now it can navigate from anywhere in this room to anywhere in this room. All it has never done this before. And it knows the right relationship between all of them. It has figured out all the correct transitive relationships by learning this. So it's not just a one-dimensional is A greater than C. It's a two-dimensional relationship it has figured out. Does that make sense? So I can create a one-dimensional version of it. It will be too simple to solve. But now it has actually figured out what are the relationships in 2D between all these different things. And you can ask that question in this graph and answer them. Does that make sense?
I think it does. But typically I think we're used to thinking of transitive inference problems outside of that kind of a graph.
Correct. Well, yes.
Where only
right.
certain elements are sampled. But I think
right.
I can. So the idea there be that there will be shared latent states here or contexts that are connecting them.
Correct.
Through
yes, exactly. Right. So remember that the experimental setting does not use any information about these are rooms. These are just some sequential observations coming in. So it is almost like I saw A after B, and then I saw B after C under some action, right? And then and in this case, it's like when I take left from or right from A, I reach B. When I take right from B, I reach C. So these are two disjoint episodes that I saw. And I might see another one when I take right from C, I reach D. These are three different disjoint episodes I saw. And transitive inference is about being able to put them together. And in typical places in which people attack this, there is no aliasing. A is a distinct observation, B is a distinct observation, etc. In that setting, it's almost like our model will solve it trivially. But it becomes more interesting where you not only have to observe things, but also put them in a latent space. And the transitive relations are actually in the latent space, not in the observed space, which is what is happening here. If you remain in the observation space, the transitive relations are just hard. But
indeed, and that's usually how it's done and experimentally, I think, outside of space.
Correct. And here I'm just using space as a visualization, right? I could move these things arbitrarily apart. It's just the relations between them.
I see. Thank you.
That raises this other question that I was going to ask about one of the key things that we're, working with theoretically is this idea that like the parietal lobe encodes kind of this abstract latent space that we're talking about, kind of independent of sensory features, right?
Yeah.
And then the ventral pathway encodes like the sensory features. So you have the separation between structure and content.
Right.
And I'm not clear in this framework whether there is such a separation, or if there is, how that works.
Yeah, there is. In fact, I was thinking about this. Basically, the latent space here is the abstract space, and you can rewire that latent space to a different set of observations.
Okay.
So remember, there is an emission matrix connecting the latent space to the observation space, and now the wirings in the latent space can be thought of as a schema, and basically you can make that schema rewire to another observation space.
Okay, but you'd have to like do some significant surgery to do that.
No,
actually like rewire connections from those new input guys. Like, how would that actually work?
Can you just have clones?
Right.
Clones in the emission matrix also. Is that
clones
possible?
in the emission matrix. I was just thinking about that the other day. Basically, saying that you maintain multiple emission matrices.
I see.
And you can rewire them. We have some experiments in the paper about how this rewiring happens, but we haven't thought too much about the neurobiology of it. But that's a very interesting one to think about. But I think, in fact, we had published another paper called Schema Networks in 2017, which was about schema formation and how do you play games using schema formation. And the funny thing is that although we arrived from two different places for these two papers, the method of creating latent states is very similar in both of them. It's this cloning mechanism. But both of them were arrived at from one we came from Drescher's schema work to get there. And he calls it synthetic items. You create this extra states called synthetic items, but those are exactly the same things we are calling clones. And so we hope to be able to merge these ideas together and kind of create the PFC schema equivalent sometime in the future. Great question. Okay, so let me.
I have a question too.
Yeah.
So I mean, humans are okay, but not amazing at transitive inference, depending on the task, I guess. But so like in your model, like how does it fail when it fails? Like, do the two clones get activated and don't find each other, or does only one clone get activated, or what do you think?
Usually it fails by not separating the things enough. So if
the prior, okay.
So that's how it fails. I hope I can show you some examples of a failed graph learning somewhere so basically if I increase the amount of degeneracy in this room, it will start becoming imperfect. It'll start merging some portions of this overlap with some portions of this overlap, etc. So some places in this overlap it will lose track. Oh, am I in this room or that room? But then maybe it takes another step and I'm in this room. So those kinds of things will start happening.
Cool.
Okay, so this is the classic Muller and QB experiment. This was done over last weekend, so I haven't delved deep into it. So this is our application of that. It's a circular room, so it's circularly symmetric. There are just 4 distinct observations. So there's a boundary observation here. There's an interior. There's another interior, and here is the white cue card in one corner. That's the experiment setting there. So remember, there are just 4 observations. So the model is ever seeing. Is it 1, 2, 3, or 4? That's all it is seeing, and it's actions, right? That nothing else. No locations are being sensed, and this is the graph it learns. I haven't put in the color mapping, but this is the graph it learns. But you can map. You can take each of these neurons and plot their place fields, and here is an assortment of some of them. So you can see that it has learned to parcellate out the space into kind of contiguous clumps. Each neuron is basically taking up each latent state is here taking up the responsibility for modeling part of the space. And so now we can do the classic Muller and QB experiment. Here is a place field of a neuron. This is clone number 122. I can basically say, hey, I want to put an obstruction in the field, which is like this, and what will happen to this field? So this is the line. In fact, all these places are still, this obstruction is places that these places are still visitable. It's not like the obstruction is along this diagonal. So these places are still can be visited by the rat. And the result is that, yeah, the place field will vanish because, you are breaking the context. So it is not too surprising that the place field kind of disintegrates. And it also, even if you put a transparent one, and in our case, transparent one means that the rat is allowed to visit all the states. It's just that when it is in this state, it is not allowed to move in this way. And when it is coming from this side into this state, it is not allowed to move this way. So the rat is allowed to visit all the states. No states are disallowed. And that's a transparent idea. But in both cases, because some motions are disallowed, the context doesn't get propagated. And because of that, the place field gets affected. Now, does it mean when I change the connectivity, the place field will get affected always? No. In fact, this is a very recent paper, February 2021. Hippocampal place fields encode global location, but not connectivity in a complex space. This is the takeaway from that paper. It's a very cool experiment, but I'm going to argue that it's a wrong takeaway. Here is this is their setting. There are 4 identical rooms, but there are enough distal cues such that the rooms can be learned without repetition of the place fields. So the way I set up this one, so you can see that the peripheral observations are identical in all the 4 rooms. The center is uniform, but I have put different observations in the center corresponding to the different rooms. So this room has some 112 different locations or something. The total number of disjoint observations I have is 25. So it's almost like 4x underrepresented in terms of the amount of degeneracy. And if I let the rat run in this room, yeah, it will learn this graph, and it learns it without any field repetition. In fact, you can see that here it messed up. You see that because remember this, when the rat is here or here or here or here, it doesn't know where exactly it is other than from the context of where it came from. And so it can wire some of these things. So it can take this portion and wire it back to here, because the only way of disambiguating is to create the latent states. So here this one, it will go away if I run it long enough. I was just running quick experiments, but this is an example of an imperfection that happens. So it learns the layout, and now I can poke around these neurons and plot the place fields. So here are the place fields. You can see that most of them are learned without repetition. And this, so you can see, look at this place field. It's basically saying it lights up when it is here, but it lights up a little bit when it is here, lights up a little bit when it's here, and lights again in the corresponding locations in the other places. Now, if I plot this in, I'm plotting this in log domain. That's why you're able to see this. If I plot this in the normal domain, these things will kind of vanish into the background. It will look very, very faint. So I'm amplifying that repetition by just, but there is no degeneracy in the sense that there is a neuron representing this portion here, and there is another neuron representing this one. You can see that here, these all neurons have the same repetition field, but this 415 is active strongly here. 403 is active strongly here. So there is no, the room is recovered almost perfectly. Now I want you to pay attention to 421 and 453. So these 2 neurons, they correspond to these 2 things in the space. Now I'm going to close this door. So the door is closed now. Okay, so that means the rat cannot move across this place. So I'm doing the surgery in both places. Inside the rat's mind, I am closing that door, and I'm also closing that door in the environment so that the rat actually cannot move there. And now if I do that and do the same place field measurement, door open, door closed, it will come out to be almost roughly the same. There is no change in the place fields. Because the only thing changed was a little bit of context change in here. It doesn't matter because that location, when rat reaches this particular location, the rat knows where it is, where it came from, based on where it came from. So the place field shouldn't change. So, so both these things are correct. Place field can change, place field need not change when there is a connectivity change. And it depends on how the context is arranged around that place. So I would say my takeaway from this one is that hippocampal place cells encode context via their connectivity. So it's not that, connectivity is not there in CA1 or anything. That will be the wrong takeaway. These activations can correspond to global locations in some settings, which is in this experiment, but connectivity change need not affect the contextual response. So that's the takeaway from that one, not the one in the title of the paper. So whether the connectivity of the environment changes the place fields depends purely on the arrangement of the context in that, some situations it will change, some situations it doesn't change. And our model makes good predictions about which are the situations. So this one should be fairly easy now, splitter cells, it's just sequence learning. If you are, the rat takes two different routes, it takes the same arm, but it's going to, turn left or right. It will learn two different copies of the states, one corresponding to turning left, one corresponding to turning right. That is fairly obvious now. And here, keep in mind that this is not random walk. The rat is taking prototypical sequences in the thing, and that's why it is. If it is the rat is doing random walk, this split won't happen. And the experiments just also point this out. It is the rat is not doing random walk. It's taking particular sequences of running through the maze. And in fact, you can make it even harder. Here the rat just has a directional sense here. It's basically this place is stochastic. This place is stochastic, and there is there's a lot of overlap between these two paths. And even then, the model is able to recover the magenta paths separate from the green paths, and it will create different clones along these things for the magenta paths and the green paths. Yeah.
That's very interesting. Thanks. Could you go back one slide?
Yeah.
As I understand it, something. So I think this is really intuitive. Something interesting that I think has been observed is that when these different paths are traversed separately, there's evidence, for example, in hippocampal replay, of stitching together two separately traversed paths. So like that GJK with the IH. So if those are encoded as different contexts, that's great. That implies some sort of structural inference.
Yeah, it is great because it's basically saying, hey, look, this arrangement of clone structured thing is a great way to explore structural generalization. Basically, even though these are in two clones, they are connected by the same observation, right? So at any point in my context, I can basically say, hey, look, what if I forget my past and I just anchor onto the current observation? Then both those things will be equally active. And those are great structural explorations you want to do if you want to do memory consolidation, some kind of reasoning, all these things.
So then what is the mechanism that tells the model when you would want to do that? Is there?
Yeah. One is you are totally surprised. You are screwed. Basically saying, oh, look, I could not predict the next thing. What I don't want to do is I don't want to carry the wrong context around. What I want to do is I want a reset. And basically saying, look, I am starting fresh from here. And this reset, you might actually, and this is related to smoothing and some idea like Neserne in n-grams. So let's say I'm walking around, and I start getting confused, and now I'm like totally lost. And now you might say, oh, I want to reset my thing right now, right? But what actually you should do is that you should reset the thing probably a few steps in the past rather than cutting the context upfront here, right? Because you had your confusion might have happened sometime back in the thing, and it was trying to propagate this wrong context forward, but the actual oh, this total failure happened only a few steps ahead. So this reset can be in the current step or few steps back in the path. In fact, there is the right way to do these resets. on, how do you keep alive the thing that, oh, you might be walking on the path, or that you might have switched out of that path and you might have started walking some other path. How do you keep alive these different contexts, and how do you reset between them? There is some interesting way of doing that, this kind of smoothing. But the important thing is that this clone structured organization gives you a good mechanism for doing many variations of that. And there are many variations you can figure out based on what is the problem that you're trying to solve. Does that
right? So it seems so that makes sense to me. But I also, I suppose something we've been thinking about a lot is the role of goals.
Yeah.
So it sounded like you were touching on that at the end.
Yeah. So
depending on
so if you're trying to so basically this is related to planning. How do you plan using the CSCG, right? And when you're doing planning, you are propagating multiple paths at the same time, at least in our model. So basically, let's say I'm here, and I want to go forward. Okay, in this particular case, it will still propagate only one path. Let's say, but I am uncertain about where I am. So let's say I just entered this position, and I think I am in this red sequence with 0.9% probability, but I think I am here with a 0.1 probability. This happens in real life, of course, where there are many situations are confusing. So then in that case, you are propagating, when you're planning, you're propagating both things at the same time. And then you want to evaluate the trade-offs between, look, if I am here with 0.9 probability, but I'm going to get $1 million when I get to this end, versus if I am here, but I'm only going to, I am certain about my position, but my reward is small, but I am uncertain about my position, my reward is big. You want to evaluate both those options and decide which way to go. So you want to basically propagate all the options forward, see what the evaluations are, and then make a decision based on that. And when you're replaying things based on that, if that is what you're trying to do, then you want to replay all the paths forward simultaneously.
It's an interesting prediction.
Yeah. So it will be basically, if this graph exists, there are many different ways to control inference in this graph, and many different questions that can be asked in this graph. So you can ask, what is the quickest path I want to take? You can ask, what is the most rewarding path you want to take? You want to minimize the amount of danger. All of those things are different questions that can be asked on the same graph. And depending on that question, or you want to say, oh, where did I come from? That's another question. And depending on those questions, you want to have different replay mechanisms in the graph, and those I think will be controlled by PFC. Many of those things. And that interaction will be something very cool to figure out. All right, keep going here. So the lab cells. So this is also interesting. Remember, this is Chensen's experiment where the rat is running labs like this for lab 1, lab 2, lab 3, lab 4. It's running in the same maze. So it's basically getting observations 1, 2, 3, 4, 5, 6, 7, 8. Then again repeats the same observation. Then again repeats. At the end of the 4th lab, it is rewarded. And what will happen in the CSCG? It will create different clones for the different labs. When it goes the second round, it will be a different clone of the observations. And then if you run inference, this is what it will look like. The correct clone will be fully active.
Sorry, Dilip, can I just quickly, why is it creating different clones for the different labs?
Oh, because at the end of the 4th lab, there is a different observation. So there is at the end of the 4th lab, there is a reward.
Okay.
So that means it needs to keep track of which lab it is in to be able to make that right prediction at the last.
So the model generates clones of labs 1 through 4.
Correct.
After you get rewarded on the 4th.
Correct.
And that's because it's basically treating laps 1, 2, and 3 and 4. The initial assumption is it's all the same sequence. And now you've created a new sequence. So would you just create two? Why create Why create 4 clones instead of 2? Because to the animal, 1, 2, and 3 are exactly the same, and it's only the 4th that's different.
So in this case, the way we have set up the experiment is it's basically there is no reward or anything in this experiment. It's more like set up like there's a start, then 1, 2, 3, 4 up to 12, 1, 2, 3, 4 up to 12, 1, 2, 3, 4 up to 12, and 4 times, and the last one a state called end state. Okay, so basically 12. There are just 14 states. So 0, 1 to 12, 1 to 12, 1 to 12, 4 times, and state 13. Oh, sorry, observation 13. That's the input sequence. And when that is given to the model, if it doesn't create 4 clones, it won't be able to predict the occurrence of that last thing correctly. It will make errors every time, because if it created only 2 clones, at the end of the second lap, it will not know whether it is going to see 13 or 1. So if it has to be completely certain it is going to see, at the end of the second lap, it is going to see 1, it needs to create another clone. That makes sense, right?
That makes sense. Yeah, I'm still kind of struggling as to, essentially the model thinks that lap 1, 2, and 3 are 3 repetitions of the same sequence, right? Or does it think 2 and 3 are repetitions of the same sequence, and 1 and 4 are different because you have start and goal?
Well, it just came sort. Let me show the graph of how the it's
Okay.
too much to say. What the model thinks. It's basically this is how the model will wire up, right? It's basically saying observation 1, it will wire to this one, then and then this observation 12 will wire to the next clone of observation 1, and this is how it will just wire up if I give this input sequence to the thing. So it's hard to know what model thinks, but I can show you from evidence what the model thinks. Basically, now what happened is that if I give after the start state should have been shown in this one. So this clone is wired to also the start state, and this clone is wired to the end state. That should have been in this picture, but you can just fill that in. So when after the start state 1 comes in, this is the observation. Observation is fed to all the clones, but this clone will be active the most because that's the one connected to the start state. And then when 1 comes around again in the next lap, this clone will be most active because that is the one corresponds to the this lap ending and connecting. The other clones will be partially active because of the smoothing, but the correct clone will be, so in that way the model will think that it is in the second lap and the third lap, etc. So so that's why the correct lap activation will be strong, and the incorrect laps corresponding traces will be slightly active, a little bit active, and that's exactly what Chen-Sun's paper shows. The correct lap is active, but there are these other traces which are active, and these are the clones corresponding to so these are the correct clones. These are the incorrect clones in some ways. And this partial activation is important because if you do some surgeries of the room, if there are noise, etc., you want to recover. So you need to keep the partial activation alive. And one example of doing a surgery is expanding the room and then basically saying, okay, now the rat, when it is at 9, it should be seeing 8, but it sees 9. So the rat has to reset its internal thinking of where it is. So basically, none of those changes affect the model much. It does the right thing in terms of propagating the context forward. This is the one observed in the model, and I think the paper also basically showed that okay, these kinds of event-specific representations carry on even if you change the length of the lap. And also, this is the experiment that we haven't done, but I can predict what will happen. It will happen same as what they observed in the paper. Suppose I don't reward at the 4th lap. I reward now at the 5th lap. After having trained on 4 laps, I train on the 5th lap. What will happen? It's very easy to understand from what we have seen so far. At the end of the 4th lap, it did not see the reward. It will reset its mind, thinking that, oh, I am not on the 4th lap. I should just keep going forward, and so the activations will shift. That's exactly what they saw in the paper. We haven't done this experiment, but I can kind of predict that, okay, that's what will happen in the model.
Got it.
Yep.
Okay, I'm sorry, I have to go, but we'll thank you so much for doing this. I really appreciate it.
Thanks, Darren. Thanks for having me. And I think I'll be done 3 slides down. And so you can see the recording.
Okay, terrific. Thank you. I hope we get to meet up in person at some point.
Yeah, I hope so too. I'm waiting for the pandemic to be over. And yeah, that'll be great.
Terrific. All right. Thank you so much. Bye.
Yeah, thanks, Darren. Okay, so so another thing is that this representation allows for learning hierarchies. You might have seen these papers where SR is used to learn hierarchies, but there are a few things there. One, SR is not needed. You can just learn the if you just take this graph and learn just the Markov chain. Each node is a different observation. Each node is a different number. If you just learn the Markov chain and do community detection, it will discover the hierarchies. So you don't actually need SR to discover the hierarchies. Second is that all these nodes are assumed to have distinct observation in that work. What if these nodes are not distinct observations? So in this case, look, this node, and this node, they are emitting the same color of the observation. So if you just learn a first order Markov chain or SR on this one, there is no community that will be discovered. Whereas in our model, so if you take this one, do SR or first order Markov chain, you'll learn this graph, no community will be discovered. If you use CSCG, it learns the correct latent things, and then you can do community detection on this one to discover the correct communities on the latent graph. And that can be used for hierarchical planning, etc. We have some things in the paper related. That again, it's done using message passing in a hierarchy. You use the top level of the hierarchy to inform how to do message passing on the lower level of the hierarchy. And these are all things that could be handled through the PFC. Probably not, it's not the hippocampus probably doing it, it's probably the PFC that is doing it. I'll make a few observations about replay, and then I will conclude. So we did not do anything specific to replay in the paper, but I think it is compatible with many of the observations that we see regarding replay. So there are 2 places where replay will be used in the paper. One is runtime replay. When the agent is behaving in the world, it needs to do replay for basically thinking forward and backward. And that thinking can be any kinds of thinking. Where do I want to go? Is it advantageous to go there versus the other one? Where did I come from? All those things are thinking, and all of them require some form of replay, whether it can be propagating forward one trajectory or multiple trajectories in parallel, etc. But all of them are compatible with the model. One prediction is that you need different sets of neurons to propagate forward and backward, because this is actually implemented in neurobiology, right? Not using in a computer science model. I can take neuron A connects to neuron B, and I can of course walk backward from neuron B to neuron A. But in neurobiology, you probably need separate set of neurons to be able to do that. So that's one prediction from the model, that you need different sets of neurons to do forward and backward, which I think is, it's not a prediction specifically from our model. I would say I think it's just a fact of neurons being unidirectional. They're not bidirectional things. And then the other part is replay for memory consolidation. This is already used in the model. I just did not bring it out. And this one needs forward and backward replay. And let me explain what this means. If I showed all these graphs to you, right? Of this is what the model learns. But I was partially lying, because if I just do the soft EM algorithm, which is used for doing the HMM learning, the graph that ends up is like this. It will have lots of, basically none of the clones will disappear. All the clones will be used, but in a very diffused way. And there will be lots of weak connections. This is where I'm saying that, look, if I take a clone and spread it into 5 different clones and change the corresponding inputs and the corresponding outputs, nothing changes, right? There is no pressure in the model to collapse that. it can keep those things around. So we do this additional thing called Viterbi consolidation, where after having learned the model using these soft updates, we basically say now try to collapse these things. Now instead of learning using uncertainty, you're basically saying you're trying to decode a path through the model, and you will use that decoding. So you can think of this as soft EM versus hard EM. In the soft learning, you are keeping all these uncertainties alive and propagating. In the hard EM, you're basically saying, look, I'm not keeping all the uncertainties alive. I just want to decode which state I am in, and then use that to update the graph. So if you do that, so that requires both forward and backward propagation to the graph. And so this is this function we have learned Viterbi. So it takes this graph as the input. This is non-consolidated graph. So it does forward and backward replay using hard decisions along the route, and then it gets this graph. So this is memory consolidation, where now lots of neurons that were occupied earlier will be disconnected. It will some connections which are diffuse will get strengthened. So this is learning time or sleep time replay, I would say, which is used for memory consolidation, which is already used in the model. We just did not pull it out as a separate thing. Some differences compared to other models, very simple in principle, not much hard coding. You can see it's a very, very simple principle just being used. And it also actually performs the task. In all these things, it's performing the task of doing the prediction, planning, filling in. It can navigate all those things. It's not just showing the receptive fields. It's actually being able to do the task. And again, if you now go back and read some of Eichenbaum's papers, Busaki papers, etc., you will see the breadcrumbs of this idea there. Eichenbaum talks about this memory space. There is a node here, etc., but he doesn't get very precise on what that node is. There is this overlapping node, but he kind of mentions that you need 2 copies of them, but he doesn't get really precise. Here too, this was the paper that when I saw, I jumped out and say, oh, wow, okay, this is corresponding to what we are thinking. Where you can see that, look, there is 2 copies of, CD, BC, AB, but there are 2 copies of B, 2 copies of C in this model. He doesn't get quite precise about it, but now, if you go back and read those papers in the context of this model, I think it will become clear what he was thinking. And also this paper from Yuri Busaki about how to think about hippocampus as just sequences. And this was this hit the nail on the head on how to. So that is what you're seeing is just a bunch of co-occurrences with no particular relationship between them, and you're just sequencing them. And that's exactly the way I think about it now. So I hope you will now agree that all these disparate phenomena are the same, and that space is a sequence. That's the end of the paper. I want to conclude by advertising another paper. This is, if you are also interested in the cortex beyond hippocampus, this is a paper we put online just a few, I think 2 months back. And there are some connections between the cloning idea, which is used in the cognitive maps paper, with the cortical column representations in that paper. So it might be, if you're interested, you might want to look at it. For more information, you can contact me anytime. And here are some of the papers. And you can also follow me on Twitter, where I am sometimes active. Thank you for listening.
Yeah, thank you so much. That was really interesting. Yeah, I think we've been having conversations in the group about, very related ideas about, that the graph is really the underlying structure behind space. And I showed a diagram very similar to that. So I think we're definitely on the same page in terms of this is the underlying kind of representation.
Great, awesome. Yeah. That's good.
Yeah.
Thanks. Any further questions?
Can I ask a question? So in terms of the difference between the successive representation, so you showed that first thing where the maze, there's the 4 rooms and you block with a door.
Mm-hmm. Yeah.
So in that case, the successor representation, it would I guess it would it would lose that 4 rooms thing. It would be like one long. It would be like one long.
You're talking about this one, right?
Yeah.
So this is the problem with successor representation. It's, if you are assuming the location is being sensed, that is it's basically, you have to ask the question, what is the input to the successor representation? Is it a location as in X and Y, or is it an observation from the room? So if it is an observation from the room, like, if I basically say, look, this particular setting has only 24 observations. So that means if I learn a successor representation on these observations, it will be a 24 by 24 matrix, which does not represent this room, because the room has close to 120 different locations in it. So it won't be able to separate the rooms, and it'll be some weird mishmash of rooms and boundaries, etc. If the successor representation's input is location, which it should not be, because who is giving the location? There is no GPS in the brain to say I am at XY location. So if the successor representation's input is the location, then of course, what you're inputting to the successor representation is this graph, the perfect version of this graph, because each node is the input. And what does successor representation do after getting this graph as the input? It blurs the graph, right? It's basically saying, look, if I am here, you tell me I'm going to go here, but look, I want to average out all my future things. So it takes this graph as the input and outputs a blurred graph. That's what successor representation does. And that blurred graph will predict that, okay, if I change the connectivity, the blurring will be different, right? So that will predict that the place will be changed. So that's the difference.
Yeah, that makes total sense. So it's basically like the successor representation, it can't handle, like, I guess, collisions, like where the observation is the same, but
correct.
Actually in a different place. It cannot handle
degenerate observations. Correct. Exactly. Yeah.
And so
and that is the crux of the problem, not the because if this graph is the input to the model, you think of it like, suppose you give this graph as the input to the model, like almost all navigation problems can be solved efficiently in this graph using message passing. So there is no need to blur this graph to do planning in this graph.
Okay, that makes sense. Thanks.
Any further question?
Thanks for a really nice talk. Something we've been interested in is this idea of structural inference in the context of some of our studies, and that relates to what Randy was talking about. This idea of sort of abstracting a structural representation away from a content-based representation. And so in our lab, something we've been thinking about is that often those sequences on a graph are not actually experienced in a sequence. That they might be in this context of actually moving around a graph, like an animal in a room, and or in the real world. But many of the problems that we sample are sampled discretely and out of order. So I was curious if you thought about that problem and sort of whether you envision this kind of model also being applied to that more abstracted set of problems, or whether you think that sort of parietal or prefrontal or whatever entorhinal kind of structural inference is happening, and have thought about the utility of it within your framework or a different model.
So we are thinking about structural inference quite a lot, and basically how to extract schemas from this one, and quite a bit of, so for example, if the episodes are disjoint, then as I showed in that thing, the model can connect many of these disjoint episodes. I want to show something even simpler So one kind of structural inference that the model enables is just because of the clone structure. Let me see that. Okay, suppose we were just memorizing the sequences by allocating a specific clone to them, right? So basically suppose I saw the sequence C, A, and B, and I just allocated specific clones to them as a pure memorization mechanism. And at a different time, I saw A followed by C, followed by E, right? Now, I can do a structural generalization between them using the idea that these are all clones of each other. Somehow erase from your mind these connections. And suppose I think I learned AB in one episode, so I wired this neuron, this clone of A to this one. I did AB. And when I saw BC, which is at a different time, I wired this clone of to this one. So it'll be AB here, BC here, and then I say CD here. I can still, even though I did not do any consolidation between them and try to connect them, I can still basically say, if I want to do structural inference based on them, I want to relax my assumptions. And one kind of relaxation I will do is basically say, when I'm at B, what are the other things I should start? Which is basically saying, oh, the other clones should be the ones I should start. And so basically a form of smoothing on this graph allows you some structured inference already.
Right.
So that's one. And the other one was basically saying that, look, I can take these schemas and connect it to a different set of observations. So basically, suppose I learned a pattern like this. It is basically the pattern is X, X, Y, 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 Z, 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 or Z, 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 Z. So basically 2 repetitions of the first observations, 3 repetitions of the second observations, 4 repetitions of the third observation. That's a sequence I learned. That particular pattern can be thought of as a schema and rewired to some another set of observations. Basically now say, is it similar to AA, BBB, CCC, or is it similar to II, JJ, J, KKK? So you can take the same schemas and wire it to different set of observations. And that's something that we are looking at. I don't think, basically what I told you is the level of depth I have gone into it. But it's a very interesting problem to work on. Yeah.
Thanks very much.
Cool. I should run now. There are other meetings. So thanks a lot. This was super interesting. Thanks a lot for having me. And
that's really great. Yeah, thank you so much. Yep.
Thank you. And I hope I can, if the recording went well, I can get the recording.
Yeah, I'll send you a link. Yep.
Okay,
yep.
Great.
Okay.
Thank you so much.
Great.
Okay.
All right.
Bye. Take care.
Bye. Okay, and I don't know what's up for our schedule going forward, but certainly if anybody knows of other guest folks like that we could give talks, that's very fun to hear about that stuff. So keep that in mind.
I don't know if this is the right forum for it, and not everybody is here. I think who would be relevant, but I remember when Miriam and Jake and others came and presented in our lab meeting, you mentioned the model you were working on with, I think, Xiaonan, and
yes,
on integration.
Yeah, right, during sleep kind of thing. Yep.
Episodes.
Yeah.
Is that sort of in a state where it would be interesting to present or help with feedback? I mean, we'd certainly be interested in learning about.
Yeah. I guess Shannon's not on right now, but he's theoretically writing that up pretty soon. I wrote a kind of intro to the paper that lays out some of the ideas, but it would be good, I think, to try to maybe reinstate some of that and maybe get some progress on that. So that would be a good idea, actually. Yeah.
if we're talking about the next week thing, I was actually the one have to present next week.
Oh, okay,
Right.
great.
Yeah.
Did I forget we had some kind of schedule that we made, roughly speaking.
But yeah, that was scheduled.
Great. So do you does that still work?
Yeah, I mean, I can present.
Great. Cool. Yeah, so I don't see that schedule zooming through here on my list, but we can kind of iterate from there. I gotta actually get going at this point, but great. So Alex, we'll hear from you next week, and then we'll see about getting Sean on the schedule as well. And then yeah, any other ideas anybody has? Great to talk about these things. Okay.
Sounds good.
All right. See you, everybody.
See you, everyone.
See you.